Okay, in this lecture we're going to begin by talking about how the Y chromosome makes a male in humans. So one of the ways that we have proven this is by looking at conditions in which something has gone awry with the Y chromosome, or the X chromosomes for that matter. So when we look at a human karyotype, we know that there are 22 pairs of autosomes and 2 sex chromosomes, and we know that males have the X and the Y chromosomes and females have X and X. So let's look at what happens when you get differences. Right, so like I said, females have 2 X, males have 1 X and 1 Y. All right, so and like I said, we've determined that the presence of the Y chromosome is what makes a male a male. Now, how do we know this? Because remember, in the nematodes and in the butterflies, it was the fact that you only have 1 X chromosome that made a male and not, anything else. So how do we know that it's not the fact that males only have 1 X chromosome, and how do we know that it's the Y chromosome that's making the difference? So the way we figured this out is by studying individuals with abnormal chromosome numbers with sex, within their sex chromosomes. So we're going to look at 2 syndromes, Klinefelter syndrome and Turner syndrome. Now, both of these syndromes result from non-disjunction of the sex chromosomes during meiosis. And we'll talk more about non-disjunction in Chapter 8 when we talk about chromosomal abnormalities. All right, so in Klinefelter syndrome, the individual has 2 X chromosomes and 1 Y chromosome. So they have 47 chromosomes total, which basically just means they have an extra X chromosome. They are phenotypically male. They do have a penis and testes. They do have other symptoms, so they're usually abnormally tall, have long arms and legs. They're usually sterile. They have some feminine development, so they'll have kind of like the rounded hips and a little bit of breast development. And they may have subnormal intelligence, but they are still considered male. Now, people with Turner syndrome, this individual only has 1 X chromosome instead of 2. They're phenotypically female. They have female genitalia. They have ovaries, but they're underdeveloped, so they're sterile. They have underdeveloped breast development. They also may have cognitive impairment. They're typically short and have skin folds at the back of the neck, but they are phenotypically female. So far we've seen someone with 2 X chromosomes, like a normal female would have, and an additional Y chromosome is male, whereas someone that only has 1 X chromosome and no Y chromosome is female. So that shows that the Y chromosome is what determines maleness. Now, some other syndromes, triplo X syndrome, called a metafemale. This actually is when you have 3 X chromosomes. These are phenotypically normal female. They may not even be aware that they have the condition. Occasionally, they may have underdeveloped sexual characteristics. They may be sterile. They may have some mental issues, but sometimes they're completely normal. And a situation where you have 2 Y chromosomes, called a supermale. These people are typically above average height. They may or may not have subnormal intelligence. In some cases, they have personality disorders. And in fact, they did a study and found in prisons that there were a large percentage of the people that had the XYY condition were in prison. So at one point, they thought that something about the extra Y chromosome made them more aggressive or have personality disorders that would lead them to be criminal in nature, but there's no proof to really show that. There's plenty of people that have 2 Y chromosomes that are completely fine in society and are not criminals. All All right, so let's talk just a little bit about non-disjunction. So these conditions like Klinefelter and Turner syndrome are caused by non-disjunction of the homologous chromosomes during gamete formation. So during meiosis, when the X and the Y are synapsed together, they don't get pulled apart during anaphase. And so you have these sperm that carry both an X and a Y. And so if they're fertilized, if they fertilize an egg, you can have an XYY or an XXY, depending on where the non-disjunction occurred. Like I said, we're going to talk about this more in Chapter 8. Okay, so is there such a thing as a 45Y syndrome where you don't have an X chromosome? This could theoretically happen during fertilization. An individual could be produced that has this karyotype. However, it wouldn't survive and it would be aborted shortly after conception, because lacking an X chromosome is not compatible with life, and that you wouldn't have a baby that would be born with this syndrome. All right, so let's talk a little bit about why the Y chromosome makes a male a male. So we've given evidence that shows that the Y chromosome is the one that's involved, and we know that lacking a Y chromosome will result in a female, and that the number of X chromosomes you have has no bearing on sex determination. So what we determined is female is pretty much the default setting for sex, and in the presence of Y chromosome, that changes to male. In the absence of Y chromosome, it remains a female. So let's talk a little bit about embryonic development. Very, very early on, when we just look like little beans, you can't differentiate between males and females. We're what we call potentially hermaphroditic, because we have bipotential gonads. We actually have it kind of set up where you can go either way. By the 5th week of development, we have these gonadal ridges develop, and the outer portion, which is called the cortex, can develop into the ovaries, or the inner portion, which is called the medulla, can develop into the testes. And so they're both there, ready for development. And it's which genes get expressed determine which one is going to continue to develop and which one is going to just kind of disintegrate. In males, what happens is if those cells have an X and a Y chromosome, and that genes on that Y chromosome are being expressed, then the testes development is initiated around week 7. The male duct system develops, and embryonic testicular tissue starts to produce the male hormones like testosterone that's responsible for all the secondary male characteristics. But the actual spermatocytes don't get produced until puberty. Now in females, if there's no Y chromosome, and so there aren't any genes from the Y chromosomes being expressed, then the ovarian tissues will develop, and then the female duct system develops into fallopian tubes, and around the 12th week of gestation, the actual oogonia start to develop, and primary oocytes appear. And of course, we've talked about this later. They arrest in meiosis 1, and do not continue to undergo meiosis until puberty begins. All right, so let's talk about these genes that are present on the Y chromosome that turn the switch on for male structures. We know that the Y chromosome has at least 50 genes. There are regions here, if you note on the ends of the Y chromosome, called PAR, that stands for pseudoautosomal regions. These are actually homologous to the X chromosome. And so these are what synapse during meiosis, and actually crossing over occurs in these regions between the X chromosome and the Y chromosome. But it's a very small portion of the Y chromosome. In the center here, the MSY stands for the male specific region. This region does not combine with the X chromosome. It has completely different genes on it. Notice also there's this large heterochromatin region of the Y chromosome that has no genes and is genetically inert. Now within the male specific region, there's one specific gene that's really important called the SRY, that stands for sex determining region. And it is the one gene that controls male development. So here you can see the X and the Y chromosomes actually synapsing during meiosis. So they do pair up and segregate during meiosis. Okay, so let's talk about the SRY region. If you are a male, but that one little region gets pinched off of your Y chromosome somehow, like maybe during meiosis, that piece is missing, you'll still be a female even though you have a portion of the Y chromosome in your cells. And that's because the SRY encodes for a protein called the testes determining factor. And really what this is it's a transcription factor that's going to go and turn on genes, actually it's on autosomes, that are really important for development of testes. It's kind of considered like a master switch. So when it binds to DNA regions, the big things it does is it suppresses development of the female reproductive tract and supports development of the male reproductive tract, and may be involved in continued suppression of female characteristics throughout the life of the male. Something that's interesting about males and females is, surprisingly, the ratio of human males to females is not 1.0. And I'm not just talking about in places in China where they selectively, try to have boys. Just in general, the primary sex ratio of, which is how many males to females are conceived, is about 1.2 to 1.6 to 1. So that means 120 to 160 males are conceived to every 100 females conceived. The secondary sex ratio, which is how many males to females are born, meaning they actually make it through the 9 months of gestation, is about 1.06 to 1, so about 106 males to 100 females born. Now what's interesting about that is clearly more males than females are being conceived, but a large portion of those males don't make it to birth. So why are more males than females conceived? Well, one explanation may be that because the Y chromosome is smaller than the X chromosome, that those sperm actually can swim they're a little bit lighter and can swim a little bit faster, so they just have a competitive edge. They can get to the egg quicker and fertilize it faster. But clearly the fetal mortality rate is higher for males than females, and we don't know really what the reason is for that. Okay, so we've talked a lot about the Y chromosome. Let's go back and talk about the X chromosome some. And we talked about this a little bit in chapter 4 when we talked about X-linked traits. And remember that the X chromosome has a lot of genes, especially in comparison to the Y chromosome. If you pull up the PowerPoint online, this is actually a link, and you can pull up all the genes that are on the X chromosome. So, the majority of these genes, remember, don't really have anything to do with femaleness, and are expressed in both males and females. That's how we discovered X-linked traits, because males are more likely to display those traits, like being colorblind. So, clearly, males need the X chromosome as much as females, and a lot of those genes are expressed in both individuals. So how do we compensate for the fact that females have 2 X chromosomes and males only have 1? Because we know that when it comes to other genes, like autosomes, have an extra chromosome is very detrimental, which is another thing that we'll talk about in chapter 8 when we talk about things like Down syndrome. So how is it that females can have twice as much genetic information as males and it not cause a problem? So there are what we call dosage compensation methods that are used to account for this difference. So if you look in humans, females inactivate one of their X chromosomes very early in development and only express the genes present on one chromosome. Those inactivated chromosomes are condensed down into heterochromatin, and you can actually see them in interphase cells. Now remember, we can't see DNA really in interphase cells because it's so relaxed and uncoiled, and the only time we can really see chromosomes is when they're all condensed and the cells are getting ready to divide. But that's not the case for the inactivated X chromosome because it remains condensed. And because we have these little spots that we can see, the researcher that discovered them, we named it after him, and so they're called Barr bodies. And so also they're called sex chromatin bodies. The choice of which X chromosome gets inactivated is random and occurs very early in embryonic development. So you can see here, this is a female cheek cell, and it has that little dark stained spot. That's the Barr body. Notice in a male cheek cell, you don't have one of those spots because they only have one X chromosome, so they don't have any Barr bodies. So if that's true, then in these abnormal karyotypes, like Klinefelter's, we should, in the triplet X meta female, we should see multiple Barr bodies, and that is true. That is what we see. So in normal males and in Turner females, there's no Barr bodies because there's only one X chromosome in those cells. In a normal female or in a male that has Klinefelter syndrome, you have one Barr body. In meta females, you have, and in XXXY males, you have 2 Barr bodies. And then moving up from that, if you have XXXX or XXXY, then you have 3 Barr bodies, and so on and so forth. So the rule is cells follow the n-1 rule, where n equals the number of X chromosomes. So there's always going to be, number of Barr bodies is going to be equal to whatever the total number is minus 1. So in this case, one of the reasons that these things like Klinefelter's and Turner syndrome are well tolerated is because these extra X chromosomes get inactivated, and so they don't affect the phenotype of the individual over much. Okay, however, we do know that while inactivation, X inactivation does allow these people to live fairly normal lives, they're not completely normal. Like, you can clearly tell there's something wrong with someone with Turner syndrome and that there's something wrong with someone with Klinefelter syndrome. So why is that the case? If the extra DNA is not being expressed, and how come they do have some kind of difference? And the best explanation that they can come up with is, while inactivation does occur fairly early, it doesn't occur quite early enough that it prevents, 100% the feminine characteristics in a Klinefelter's or male characteristics in a Turner. Okay, so which X chromosome gets inactivated? Well, the Lyon hypothesis, which was proposed by Mary Lyon, states that it's completely random which X chromosome gets inactivated, and that it occurs fairly early in fetal development. Once the inactivation occurs, then all descendant cells will have that same inactivation. And the evidence that supports our theory, calico cats have that mosaic appearance on their coat, the patches of black and orange and white, and that's because they are heterozygous on the X chromosome for those colors, for that yellow and that black, and so it depends on which X chromosome was inactivated in those cells as to where the patches show up. When it comes to color blindness, actually in females that are heterozygous, their eyesight's fairly normal, but if you drill down to their retinas, you can actually tell that some cells function properly and others don't, where the wrong X chromosome was inactivated. Alright, so how this works, there is a gene on the X chromosome called the X inactive specific transcript gene, and it's only active on the X chromosome that gets inactivated. The gene product is called the X inactive specific transcript, and really it's just an RNA transcript that coats and inactivates the X chromosome. And that continues to be inactivated throughout the life of the females, which is a process called imprinting. And somehow the other genes that are associated with that center have a way to kind of count how many X chromosomes are present in the cell, so it knows how many need to be inactivated. Now in Drosophila, sex determination is a little bit different. So remember, this is our favorite fruit fly. On the surface they appear the same because females have 2 X chromosomes, males have an X and a Y chromosome. However, the Y chromosome does not appear to be involved in sex determination, because when you look at the Klinefelter and Turner syndrome, they're not really called that in fruit flies, but when you look at fruit flies that are XXY and XO, the XXY flies are female and the XO flies are male. So that's opposite from what we see in humans. However, the XO flies are sterile, so while the Y chromosome may not have anything to do with maleness, it does seem to have something to do with fertility. And the sex determination in Drosophila is much like what we see in the butterfly, in that it's a ratio of X chromosomes to autosomes, and females will have that 1-to-1 ratio and males have that 1-to-2 ratio. So in this case, the maleness genes are actually present on the autosomes, and a lot of the female development genes are present on the X chromosome. Drosophila also differ from us in how they compensate for dosage. The females do have 2 copies of the X chromosome. Males only have 1, but instead of inactivating one of them, the male X-linked genes are transcribed at twice the rate of the females. Okay, the last thing we're going to talk about is temperature-dependent sex determination. So in some organisms, like crocodiles, turtles, and some lizards, sex is actually determined by environmental factors. So it depends on what temperature the eggs are incubated during embryonic development as to what sex they'll be. The temperature at which you get equal number of male and female is called the pivotal temperature, and it appears to be related to the activity of enzymes and hormones. So for example, the enzyme aromatase converts androgens like male hormones to female hormones, and it seems to be sensitive to temperature. So a thermosensitive factor mediates expression of the aromatase gene in reptiles, so if it's not at the proper temperature, that enzyme is not going to be activated. So this is 3 different cases, and this is, different species of turtles and whatnot. In this case, you can see at really high temperatures, the turtles are all female. At really low temperatures, you have all male, and in that in-between temperature is where you have both male and female. With this case, it's just the exact opposite. At really low temperatures, you have females. Really high temperatures, you have males. But again, in the center, you have both males and females. And in this case, you have a situation where either at very high or very low temperatures, you have females. In very moderate temperatures, you have males. And then kind of the in-between temperatures on either side is where you have the 50-50 ratio. Okay, so that's it for chapter 7. So this was a pretty quick chapter. It's not as much problems and stuff to figure out with this chapter, so it's kind of a nice break after chapters 4 and 5. And then the next chapter we're going to get to in chapter 8, that's where we're going to be talking about chromosomal mutations. That one will also be a little bit easier too. I think this will be an easier week. But go ahead and after you finish watching this, go ahead and answer the review questions, and then if you want, you can move on to chapter 8.